Hello students, welcome back to Rush Learning Hub. This is part 3 of our chapter, Sexual Reproduction in Flowering Plants. In the previous parts, we learned about the structure of flowers, the development of male and female gametophytes, pollination, fertilization, and the formation of seeds and fruits. In this final lesson, we will explore some important NCERT concepts that are essential for board examinations and competitive exams. We will study the different agents of pollination, understand how plants prevent self-pollination through outbreeding devices, and learn about pollen-pistil interaction. We will also discuss artificial hybridization, a technique widely used in plant breeding programs. Finally, we will understand the concepts of Apomixis, Parthenocarpy, and Polyembryony, along with their significance in plant reproduction and agriculture. Now let us study the agents of pollination. Pollination is defined as the transfer of pollen grains from the anther to the stigma of a flower. It is an essential step in sexual reproduction because fertilization cannot occur unless pollen grains reach the stigma. Plants cannot move from one place to another. Therefore, they depend on various external agents for the transfer of pollen grains. These agents may be abiotic, such as wind and water, or biotic, such as insects, birds, bats, and other animals. Wind pollination anemophily. In wind-pollinated plants, pollen grains are carried by air currents. To facilitate wind pollination, plants produce a very large number of pollen grains because many pollen grains are lost during transport. These pollen grains are usually small, light, dry, and non-sticky, allowing them to be easily carried by wind. The flowers are generally small, inconspicuous, and do not produce nectar because they do not need to attract animals. A common NCERT example is MAJ. Water pollination hydrophily. In some aquatic plants, pollination occurs through water. Water carries pollen grains from one flower to another. To prevent damage, pollen grains often possess a protective mucilaginous coating. Hydrophily is relatively rare among flowering plants. A well-known NCERT example is Vallisneria. In Vallisneria, female flowers reach the water surface, and male flowers or pollen are transported by water currents to achieve pollination. Animal pollination zoophily. Most flowering plants depend on animals for pollination. Among animals, insects are the most important pollinators. Bees, butterflies, moths, birds, and bats frequently visit flowers while searching for nectar or food. To attract these pollinators, flowers are often brightly colored, fragrant, and rich in nectar. The pollen grains in animal-pollinated flowers are generally large, sticky, and often spiny. These characteristics help them attach to the bodies of pollinators and be transported to other flowers. A common NCERT example is salvia, which is pollinated by insects. Now let us study outbreeding devices, an important concept in plant reproduction. Many flowering plants have evolved special mechanisms to prevent self-pollination and encourage cross-pollination. These mechanisms are known as outbreeding devices. Why are outbreeding devices important? Self-pollination over many generations may reduce genetic variation. Cross-pollination, on the other hand, introduces new genetic combinations and increases adaptability and survival. Therefore, plants have developed several strategies to promote cross-pollination. Let us discuss the major outbreeding devices mentioned in NCERT. 1. Dichogamy. Dichogamy is a condition in which the anthers and stigma of the same flower mature at different times. As a result, when pollen is released, the stigma is not ready to receive it, or when the stigma becomes receptive, pollen release has already occurred. This prevents self-pollination. Dichogamy occurs in two forms. The first is protandry, where the anthers mature before the stigma. The second is protogeny, where the stigma matures before the anthers. In both cases, self-pollination is reduced and cross-pollination is promoted. 2. Hercogamy. Another mechanism is hercogamy. In hercogamy, the anthers and stigma are physically separated within the flower. Because of this spatial separation, pollen grains cannot easily reach the stigma of the same flower. Thus, self-pollination becomes difficult, increasing the chances of cross-pollination. 3. Self-incompatibility. Self-incompatibility is a genetically controlled mechanism. In this case, even if pollen from the same flower reaches the stigma, it is recognized and rejected. The pollen either fails to germinate or its growth is inhibited. As a result, fertilization does not occur. This mechanism ensures that only genetically different pollen grains can successfully fertilize the ovules. 4. Unisexuality. Some plants produce unisexual flowers, meaning a flower contains either stamens or carpels, but not both. In certain species, male and female flowers occur on different plants. Since both reproductive organs are not present in the same flower, self-pollination is completely prevented. This naturally promotes cross-pollination. Conclusion. Thus, dichogamy, hercogamy, self-incompatibility, and unisexuality are important outbreeding devices that help maintain genetic diversity in plant populations. These mechanisms increase the chances of cross-pollination and contribute to the evolutionary success of flowering plants. After pollination, an important series of events takes place between the pollen grain and the pistil. This process is known as pollen-pistil interaction. The pollen-pistil interaction ensures that only compatible pollen grains are allowed to fertilize the ovules. It is a highly specific and regulated process that plays a crucial role in successful fertilization. Let us understand the steps involved. Step 1. Pollen grain lands on the stigma. The process begins when a pollen grain is transferred from the anther and reaches the stigma of a flower. The stigma acts as the receptive surface for pollen grains. However, not every pollen grain that lands on the stigma will be accepted. Step 2. Recognition of compatible pollen. The stigma is capable of recognizing whether the pollen grain is compatible or incompatible. If the pollen grain belongs to the same species and is genetically suitable, it is accepted. If it is incompatible, the stigma rejects it and prevents further development. This recognition mechanism helps maintain reproductive efficiency and prevents unwanted fertilization. Step 3. Pollen germination. Once a compatible pollen grain is accepted, it absorbs moisture and nutrients from the stigma. As a result, the pollen grain germinates and produces a tubular outgrowth known as the pollen tube. This pollen tube serves as the pathway through which male gametes travel toward the ovule. Step 4. Growth of the pollen tube. The pollen tube grows through the style toward the ovary. Its growth is guided by chemical signals produced by the ovule and surrounding tissues. Inside the pollen tube are two male gametes that are transported safely toward the embryo sac. This is one of the most remarkable examples of directed cell growth in plants. Step 5. Entry into the ovule. The pollen tube finally reaches the ovule and enters through a small opening called the micropyle. It then penetrates the embryo sac and releases the two male gametes. These male gametes will participate in the process of double fertilization, which you have already studied in earlier lessons. Significance of pollen-pistil interaction. The pollen-pistil interaction acts as a biological screening system. It ensures that only the correct pollen grain germinates and reaches the ovule. This increases the chances of successful fertilization and healthy seed development. Thus, pollen-pistil interaction is an essential link between pollination and fertilization in flowering plants. Artificial hybridization, an important technique used in plant breeding. Artificial hybridization is the process by which pollen grains from a selected male parent are deliberately transferred to the stigma of a selected female parent. The aim is to combine desirable characteristics from two different plants and produce improved offspring, known as hybrids. Plant breeders use this technique to develop crop varieties with higher yield, better quality, disease resistance, drought tolerance, and other useful traits. Let us understand the steps involved in artificial hybridization. Step 1. Selection of parents. The first step is the selection of suitable parent plants. The breeder chooses two plants that possess desirable characteristics. One plant is selected as the female parent and the other as the male parent. The goal is to combine the best traits of both parents in the offspring. Step 2. Emasculation. The next step is called emasculation. In a bisexual flower selected as the female parent, the anthers are carefully removed before they release pollen grains. This prevents self-pollination and ensures that only the desired pollen is used for fertilization. Emasculation is usually performed using forceps under careful observation. Step 3. Bagging before pollination. After emasculation, the flower is covered with a suitable paper or plastic bag. This process is called bagging. Bagging prevents unwanted pollen grains from reaching the stigma and contaminating the experiment. Step 4. Collection of pollen. Mature pollen grains are collected from the selected male parent plant. Only healthy and viable pollen grains are chosen for pollination. Step 5. Artificial pollination. When the stigma of the female flower becomes receptive, the collected pollen grains are carefully transferred onto it using a brush or other suitable method. This ensures controlled pollination between the chosen parents. Step 6. Bagging and tagging after pollination. After pollination, the flower is again covered with a bag to prevent contamination. A tag is attached containing details such as the names of the parent plants and the date of pollination. This step is known as tagging. Importance of artificial hybridization. Artificial hybridization is widely used in agriculture and plant breeding programs. It allows scientists to develop improved crop varieties with desirable characteristics and contributes significantly to food production and crop improvement. Thus, artificial hybridization is a powerful tool that enables controlled breeding and the development of superior plant varieties. Apomixis. The term apomixis literally means reproduction without fertilization. According to NCERT, apomixis is a form of asexual reproduction that mimics sexual reproduction. In this process, seeds are formed without the fusion of male and female gametes. As a result, the offspring produced are genetically identical to the parent plant. Normally, in sexual reproduction, fertilization leads to the formation of an embryo and ultimately a seed. However, in apomixis, seed formation occurs even though fertilization does not take place. Let us understand how this happens. In some plants, the egg cell develops directly into an embryo without being fertilized. Since there is no fusion of gametes, the embryo carries the same genetic makeup as the mother plant. In other cases, embryos may arise from somatic cells of the ovule, such as nucellar cells. These cells divide and develop into embryos without undergoing fertilization. Thus, Apomixis allows plants to produce seeds while completely bypassing the process of fertilization. Significance of Apomixis. Apomixis has great importance in agriculture and plant breeding. One major advantage is that it helps preserve desirable genetic traits across generations. For example, if a hybrid plant possesses high yield, disease resistance, and good quality, these characteristics can be maintained unchanged through apomictic seeds. This eliminates the need to produce hybrid seeds repeatedly every year. Apomixis also ensures uniformity among offspring because all the plants produced are genetically similar to the parent plant. For this reason, scientists are highly interested in introducing Apomixis into economically important crop plants. Examples. NCERT mentions that Apomixis occurs naturally in several species of grasses and some flowering plants. It is also observed in certain species of citrus and other plants. Conclusion. Therefore, Apomixis is the formation of seeds without fertilization. It is a special reproductive strategy that combines the advantage of seed formation with the genetic stability of asexual reproduction. Because of its agricultural importance, Apomixis is considered one of the most valuable concepts in modern plant breeding. Parthenocarpy. The term Parthenocarpy refers to the development of fruits without fertilization. Normally, fruit formation occurs after fertilization of the ovule. However, in Parthenocarpy, the ovary develops into a fruit even though fertilization does not take place. As a result, the fruits produced are usually seedless. Let us understand this process step by step. Initially, a flower develops normally and contains an ovary. Under normal conditions, pollination and fertilization lead to seed formation and fruit development. But in Parthenocarpy, the ovary begins to grow and develop into a fruit without the formation of seeds because fertilization has not occurred. Therefore, the mature fruit produced is seedless. Types of Parthenocarpy. Parthenocarpy can be of two types. The first is natural Parthenocarpy. In this case, fruits develop naturally without fertilization. Certain plant species have the ability to produce seedless fruits under normal conditions. The second is induced Parthenocarpy. Here, fruit development is artificially stimulated by applying plant growth regulators such as auxins and gibberellins. This method is commonly used in agriculture to improve fruit production. Significance of Parthenocarpy. Parthenocarpy has great economic importance. Seedless fruits are generally preferred by consumers because they are easier to eat and often have better market value. Parthenocarpy also allows fruit production even when pollination or fertilization fails due to unfavorable environmental conditions. In addition, it helps produce fruits with greater uniformity in size and quality. Examples. Some common examples of parthenocarpic fruits include *banana, *seedless grapes, *certain varieties of oranges, *some seedless tomatoes, *cucumber. These fruits are widely cultivated and have significant commercial value. Difference between Apomixis and Parthenocarpy. Students often confuse these two terms. Remember, Apomixis results in the formation of seeds without fertilization, whereas Parthenocarpy results in the formation of fruits without fertilization, and these fruits are generally seedless. This distinction is very important for board examinations. Conclusion. Thus, Parthenocarpy is the development of fruits without fertilization. It leads to the production of seedless fruits, and has great importance in horticulture and agriculture. Polyembryony. The term polyembryony refers to the development of more than one embryo within a single seed. Normally, one fertilized egg develops into one embryo. However, in some plants, multiple embryos may develop inside the same seed. This phenomenon is called polyembryony. Let us understand how this occurs. Formation of multiple embryos. After fertilization, a zygote is formed inside the ovule. This zygote normally develops into a single embryo. However, in certain plants, some cells of the nucellus surrounding the embryo sac begin to divide and develop into additional embryos. These embryos are known as nucellar embryos. As a result, the seed contains one normal zygotic embryo along with several additional nucellar embryos. Therefore, when the seed germinates, it may produce more than one seedling. Types of embryos. There are two main types of embryos present in a polyembryonic seed. The first is the zygotic embryo, which develops from the fertilized egg cell through normal sexual reproduction. The second is the nucellar embryo, which develops from nucellar tissue without fertilization. These nucellar embryos are genetically identical to the mother plant. Significance of polyembryony. Polyembryony has great practical importance in horticulture and plant breeding. Since nucellar embryos are genetically identical to the parent plant, they help preserve desirable traits. Polyembryony also increases the chances of successful seedling establishment because several embryos may develop from a single seed. This phenomenon is useful in the propagation of elite fruit varieties and helps maintain uniformity among plants. Examples. NCERT commonly mentions citrus species, such as oranges and lemons, as classic examples of polyembryony. Polyembryony is also observed in mango and certain other fruit crops. These plants often produce multiple seedlings from a single seed. Important exam point. Students should remember the difference between the three special phenomena studied in this lesson. Apomixis seed formation without fertilization. Parthenocarpy fruit formation without fertilization. Polyembryony more than one embryo develops in a single seed. This comparison is frequently asked in board examinations and competitive exams. Conclusion. Thus, polyembryony is the occurrence of multiple embryos within a single seed. It is an important natural phenomenon that contributes to plant propagation, crop improvement, and horticultural practices. Students, with this slide we come to the end of Chapter 1, Sexual Reproduction in Flowering Plants. Let us quickly revise the important concepts that we have studied throughout this chapter. We began by understanding that the flower is the reproductive unit of angiosperms. The male reproductive organ, known as the stamen, produces pollen grains, while the female reproductive organ, the pistil, contains the ovules. We then studied the formation of male and female gametophytes through microsporogenesis and megasporogenesis. Next, we learned about pollination, which is the transfer of pollen grains from the anther to the stigma. Pollination may occur through various agents such as wind, water, insects, birds, and other animals. After pollination, pollen-pistil interaction takes place. During this process, compatible pollen grains germinate on the stigma and produce a pollen tube that carries male gametes to the embryo sac. We also studied the unique phenomenon of double fertilization, which is a characteristic feature of flowering plants. One male gamete fuses with the egg cell to form the zygote, while the second male gamete fuses with the polar nuclei to form the primary endosperm nucleus. Following fertilization, the ovule develops into a seed and the ovary develops into a fruit. In this final lesson, we explored several special mechanisms of reproduction. We learned that apomixis is the formation of seeds without fertilization. Parthenocarpy is the development of fruits without fertilization, resulting in seedless fruits. And polyembryony is the occurrence of more than one embryo within a single seed. These mechanisms are not only biologically fascinating but also highly important in agriculture, horticulture, and plant breeding. Final revision formula. Remember these three key definitions. Apomixis seed without fertilization. Parthenocarpy fruit without fertilization. Polyembryony more than one embryo in a seed. If you remember these concepts clearly, you will be able to answer many NCERT, board examination, and NEET questions with confidence. Thank you for joining me throughout this chapter. Keep learning, keep growing, and keep shining. See you in the next chapter with Rush Learning Hub.